Hi, good evening. We're back with our subject on cellular metabolism, and in specific, we'll be dealing with lipid metabolism and lipid metabolism disorders and the various processes in lipid metabolism, and then we move on to protein metabolism today. But before we could go to the subject proper, let's just have a recap on the subject with Q&A as usual. We'll have about 4 to 5 Q&As. The first question would be, what are the disorders related to lipid metabolism? So we have lipid metabolism disorders such as Gauchers disease and Tay-sachs disease, which involves lipids, and lipids are fat or fat-like substances. They include oils, fatty acids, waxes, and cholesterol. If you have one of these disorders, you may not have enough enzymes to break down lipids. That's the first question. Next Q&A would be, what are the 5 lipid disorders? Now, basically, it's classified as lipoprotein disorders, where you have dyslipidemia, Fredrickson phenotype, abnormal lipoproteins. Then we have exogenous hyperlipemia, type 1, and then an increase in chylomicrons. And next will be hypercholesterolemia, type 2A, with an increase in LDL, a combined hyperlipidemia, type 2B, an increase in LDL and AMP with VLDL, remnant hyperlipemia with type 3, where we have an increased LDL, also known as beta VLDL. The next question would be, Gauchers, what are the 4 inborn disorders of lipid metabolism? Gauchers disease types 1, 2, and 3, Niemann-picks disease, Tay-sachs disease, and Fabry's disease are all diseases where those afflicted can have a disorder of the body lipid metabolism. A rarer disease concerning a disorder of the lipid metabolism are cytosterolemia, Warman's disease, Refum's disease, and cerebrotendinous xanthomatosis, right? You have the types of lipid. Those are the types of lipids would be membrane lipids, where you have phospholipids, sphingolipids, glycolipids, and glycophospholipids. Right? Just recap that again. And glycerophospholipids. The other types of lipids will be cholesterol, steroid, triacylglycerols, fatty acids, bile salts, eicosanoids, ketone bodies, right? And having answered that, we go to the next question. Just for a continuation, there are different types of inborn errors in metabolism, including maple sugar urine disease, fructose intolerance, galactosemia, and phenylketonuria. Which term describes disorders of lipid metabolism? Disorders of lipoprotein and lipid metabolism. Dyslipidemias can be classified based on the primary biochemical disturbance, such as high or low plasma. What is lipid metabolism? Lipid metabolism involves the synthesis of the structural and functional lipids, such as phospholipids, glycolipids, sphingolipids, cholesterol, prostaglandins, etc., that are characteristic of individual tissues, and the degradation of lipids to satisfy the metabolic needs of the body, example, energy production. What are lipids and disorders of lipoprotein metabolism? Disorders of lipoprotein metabolism include primary and secondary conditions that substantially increase or decrease specific circulating lipids, example, cholesterol or TGs, or lipoproteins, example, low density or high density lipoproteins. Right? If you check into this subject here, we would like more details like Disorders of lipoprotein metabolism, including primary and secondary conditions that substantially increase or decrease specific circulating lipids, such as the cholesterols or TGs, lipoproteins, low density, high density lipoproteins, etc. Their demonstration that cholesterol lowering therapy significantly reduces the clinical complications of atherosclerotic cardiovascular diseases, ASCVD, makes it important for clinicians to be familiar with the diagnosis and treatment of lipoprotein disorders. Now here we review normal protein physiology, the pathophysiology of disorders of lipoprotein metabolism, the effects of genetic and environmental factors on lipoprotein metabolism, and the clinical approaches to the diagnosis and management of lipoprotein metabolism. Right? And a little bit on lipoprotein metabolism, lipoprotein classification composition, a little continuation to yesterday's lecture. Lipoproteins are large macromolecular complexes composed of lipids and proteins that transport poorly soluble lipids, primarily TGs, cholesterol, and fat-soluble vitamins through body fluids, plasma, interstitial fluid, and lymph to and from tissues. Lipoproteins play an essential role in the absorption of dietary cholesterol, long-chain fatty acids, and fat-soluble vitamins, and the transport of TGs, cholesterol, and fat-soluble vitamins from the liver to peripheral tissues, and the transport of cholesterol from peripheral tissues to the liver and intestines. Right? And lipoproteins contain a core of hydrophobic lipids, TGs, and cholesterol esters surrounded by a shell of hydrophilic lipids, phospholipids, unesterified cholesterol, and proteins called apolipoproteins that interact with body fluids. The plasma protein are divided into 5 major classes based on their relative density. So it's called chylomicrons, very low density lipoproteins, VLDLs, intermediate density lipoproteins, IDLs, low density lipoproteins, LDLs, and high density lipoproteins, HDLs. Each lipoprotein so each lipoprotein class comprises a family of particles that have varying density, size, and protein composition. Because a lipid is less dense than water, the density of a lipoprotein particle is primarily determined by the amount of lipid per particle. Chylomicrons are the most lipid-rich and therefore least dense lipoprotein particles, whereas HDLs have the least lipids and are therefore the most dense lipoproteins. In addition to the density, lipoprotein particles can be classified according to their size, determined either by non-denaturing gel electrophoresis or by nuclear magnetic resonance profiling. There is a strong inverse relationship between density and size, with the largest particles being the most buoyant, chylomicrons, and the smallest particles being the most dense, them being the HDLs. Right? This is a figure of the VLDLs. The lipoproteins are classified by density and size, which are inversely related. HDL high density lipoprotein, IDL intermediate density lipoprotein, LDL low density lipoprotein, VLDL very low density lipoproteins are shown here. This diagram, have a look, take a screenshot as you keep going through. Right? So I said about that on that. Let's get back to the subject matter proper with the last 1 or 2 questions. Now, what do you mean by disorder of metabolism? A metabolic disorder is a disorder that negatively alters the body's processing and distribution of macronutrients such as proteins, fats, and carbohydrates. Metabolic disorders can happen when abnormal chemical reactions in the body alter the normal metabolic processes. How many metabolic disorders are there? Although each metabolic disease individually is rare, there are more than 1,300 known metabolic diseases, and collectively they represent a significant cause. Right? There are hundreds of inherited metabolic disorders caused by different genes. I think I've already mentioned a few of them. All right, just mentioning a few examples, subsets and synonyms for metabolic disorders, inborn errors in metabolism, disorders, amino acid metabolism, amino, acidaminase, maple syrup urine disease, MSUD, homocystinuria, disorders of organic acid metabolism, organic acidurias, organic acidemias, which would be methyl maloric aciduria, 3-methyl-glutathionic aciduria or Bath syndrome, glyceric aciduria, 2-hydroxyglutaric aciduria D and L forms, as dextro and levo forms, disorders of fatty acid beta oxidation, MCAD deficiency, LCHAD and VLCAD deficiency, disorders of lipid metabolism, lipid storage disorders such as gangliosidosis, Tay-sachs disease, Sandoff disease, sphingolipidosis, Fabry disease, Gauchers disease, Niemann-picks disease, Krabbe disease, mucolipidosis, mucopolysaccharidosis, mitochondrial disorders, mitochondrial cardiomyopathies, Lay disease, MELAS, MERRF, NARP, and Bath syndrome, paroxysmal disorders, Zellweger syndrome, cerebrohepatorenal syndrome, X-linked adrenoleukodystrophy, and Refsum diseases. All right, you could go refer to the National Organization for Rare Diseases for further or additional resources. So that's it about references and questions. We're going to be going into the subject matter detail on the continuation of fatty acid or fat lipid metabolism, and then on to protein metabolism. All right, continuing from yesterday's subject matter proper, we were at the synthesis of fatty acids, the biosynthesis of long fatty acids chain in animal tissues, plants, and microorganisms is not a direct reversal of fatty acids oxidation, although acetyl-CoA is the starting material. Fatty acid synthesis occurs in the cytoplasm of cells, compared to beta oxidation, which occurs inside the mitochondria. The process of fatty acid synthesis involves two regulatory steps. The first is the carboxylation of acetyl-CoA. The first is the carboxylation of acetyl-CoA in the cytosol to form malonyl-CoA. The acetyl-CoA and malonyl-CoA are transferred to ACP, acyl carrier protein, by the action of acetyl-CoA transacylase and malonyl-CoA transacylase respectively, catalyzed by the biotin-dependent acetyl-CoA in the cytosol-CoA carboxylase known to be key enzyme of fatty acid synthesis, an enzyme that transfers CO2 to substrates. This step is a rate-limiting step and therefore a very important site in the regulation of fat accumulation. If sufficient biotin is not available for carboxylation of acetyl-CoA, fatty acid synthesis will not occur. The second major point of regulation in fatty acid synthesis is the decarboxylation of the malonyl group, catalyzed by fatty acid synthase. The multi-enzymatic activity of fatty acid synthase, FASH, it regulates fatty acid synthesis in higher organisms like yeast, birds, and mammals. FAS is present in the cytosol of many tissues like kidney, brain, lung, mammary gland, and the adipose tissues. Synthesis of fatty acids by the FAS complex stops after 16 carbons have been added, palmitate, and further elongation and the addition of double bonds are carried out by other systems. The rate of fatty acid synthesis is controlled by the equilibrium between the monomeric and polymeric acetyl-CoA carboxylase. Hormones play an important role in lipid metabolism. Fatty acid synthesis is regulated by phosphorylation, dephosphorylation reactions. Insulin stimulates the dephosphorylation of acetyl-CoA carboxylase, activating fatty acid synthesis. Phosphorylation of acetyl-CoA carboxylase by hormones, epinephrine, norepinephrine, and glucagon result in the inactivation of this system, inhibiting synthesis of fatty acids from acetyl-CoA. The role of fatty acid synthesis is to supply the needs for particular fatty acids not supplied in the diet, to convert Excess dietary glucose to fatty acids. For storage, glucose is the major source of acetyl-CoA for fatty acid synthesis. Abnormalities in lipid metabolism. Obesity, where excessive deposition of fat in the depots with decreased mobilization causes obesity. Obesity is non-endocrinal causes, is always due to ingestion of more food than necessary to meet the metabolic needs of the adult. Gauchers disease, the cerebrosides are increased in brain, liver, and spleen. There is an imbalance between the synthesis and the breakdown of this lipid. Niemann-picks disease, sphingomyelins accumulate in liver, spleen, bone marrow, lung, and lymph nodes. Gangliosides of brain show degeneration. Several variants of the disease occur. The abnormality here also is an imbalance between synthesis and breakdown of the sphingomyelins. Tay-sachs disease, abnormal glycosides accumulate in brain. There is retarded development, paralysis, dementia, and blindness. The child does not usually survive beyond 2 to 4 years. Fabry's disease, abnormal galactosyl sphingolipid accumulates in many tissues. Death occurs due to cardiac or renal failure. The next topic will be protein metabolism. Proteins contain carbon, hydrogen, oxygen, nitrogen, sometimes other atoms. They form the cellular structural elements are biochemical catalysts and important regulators of gene expression. Nitrogen is essential for formation of 20 different amino acids, the building blocks of all body cells. The amount of intake of nitrogen in food, mainly as protein, will be balanced by excretion of an equal amount of nitrogen in urine in the form of urea. Uric acid, creatinine, and amino acids contribute to a minor extent, and feces, mainly unabsorbed nitrogen. The animal is said to be in nitrogen balance. Amino acids can also be converted into glucose and used for energy through gluconeogenesis. Amino acids are characterized by the presence of a terminal carboxyl group and an amino group in the alpha position. And they are connected by peptide bonds. Protein catabolism is the breakdown of proteins and into amino acids and simple derivative compounds for transport into cells through the plasma membrane, ultimately for the polymerization into new proteins via the use of ribonucleic acids, RNA, and ribosomes. The final products of protein catabolism include carbon dioxide, water, ADPase, and ammonia. There is no special storage form of protein like glycogen for carbohydrate adipose tissue. For fat, protein storage is always accompanied by tissue growth. Amino acid metabolism includes biosynthesis of amino acid degradation, conversion of the carbon skeleton of the amino acids to amphibolic intermediate urea synthesis, and the formation of a wide variety of physiologically active compounds such as serotonin, liver and kidney, and several other tissues contain DNL amino acid oxidases active in deamination. Of the DNL amino acids, as the case may be. More of the deamination occurs in the liver normally. Biosynthesis of amino acids. Biosynthesis of amino acids takes place in association with those organic compounds which occur in the environment in oxidized states such as SO2-, N2, and NO3. Environmental forms of nitrogen and sulfur are reduced to metabolically available forms and are combined with the intermediates of carbohydrates metabolism to synthesize the 20 amino acids characteristics of all living forms. The collection of free amino acids in the body blood is called amino acid pool. They represent the relation between the removal and addition of amino acids. Protein catabolism. The amino acid enters respiratory routes in two ways: deamination and transamination. Liver plays an important role in protein catabolism. Deamination, the first stage in the breakdown of amino acids, is the removal of the nitrogenous groups. As ammonia, it is a catabolic process in which the amino group is removed and convert the amino acid into ketoacids. This is known as deamination. Such deamination results from the activation of a variety of enzymes which are either oxidative or hydrolytic. Oxidative deamination, catalyzed by a group of flavin enzymes as amino acid oxidases, which are auto-oxidizable. Flavor proteins, that is the reduced F-amino FAD, is reoxidized directly by molecular oxygen, forming hydrogen peroxide H2O2 without participation of cytochromes or other electron carriers. H2O is then split to O2 and N2O by catalase, which occurs widely in tissues, especially liver, and the amino acid oxidase reactions, the amino acid is first dehydrogenated by the flavor protein of the oxidase, forming an alpha amino acid. This spontaneously adds water, then decomposes to the corresponding alpha keto acid for the loss of alpha amino nitrogen as NH3. Hydrolytic deamination. In addition to the amino acid oxidase, there are a number of the hydrolytic enzymes that deaminate specific amino acids, particularly the sulfur-containing amino acids, aspartic and glutamic acids. For example, in the non-oxidative deamination glutamic acid, the enzyme glutamic dehydrogenase removes 2 hydrogen atoms from a combination of glutamic acid and water with the NAD as coenzyme, and a keto acid is again formed. The reaction is readily reversible. Glutamic acid plus H2O in the presence of glutamic dehydrogenase in liver gives alpha ketoglutaric acid plus hydrogen plus ammonia, NH3. The ammonia liberated by deamination reactions is not allowed to accumulate but rapidly metabolized, with the result that very little escapes into the blood. That ammonia is normally 10 to 12 milligrams per 100 ml. Increase in blood ammonia is highly toxic to the central nervous system and may be fatal. Ammonia is toxic to the nervous system, and its accumulation rapidly causes death. Therefore, it must be detoxified to form a form which can be readily removed from the body. Ammonia is converted to urea in liver, which is water soluble and is readily excreted via kidneys and urine. Liver is the main site of ammonia metabolism. The steps in the synthesis of urea in the liver were elucidated by Krebs and hence late. After deamination, carbon atoms of degraded amino acids emerge in major metabolic intermediates that can be converted into glucose or be oxidized by the citric acid cycle. To economize the metabolic conversion, this carbon skeleton of 20 diverse amino acids are funneled into only 7 molecules: pyruvate, acetyl-CoA, acetoacetyl-CoA, alpha-ketoglutarate, succinyl-CoA, fumarate, and oxaloacetate. On the basis of the amphipolic fates of these carbon skeletons, amino acids are classified into 3 types: purely glycogenic, purely ketogenic, and glucogenic. Transamination, transfer amino groups. The process involves the transfer amino group NH2 from a donor amino acid to the recipient keto acid to form the analogous amino acid and to produce the keto acid from the original amino donor. The donor amino acid thus becomes a keto acid and the recipient keto acid becomes an amino acid. The keto acids so formed are normal participants of glycolysis or Krebs cycle. An example of transamination is the formation of the amino acid by the transfer of the amino group from glutamic acid to pyruvic acid, in the course of which alpha-ketoglutarate acid is produced. The enzyme involved are transaminases or aminotransferases. Enzymes called transaminases or aminotransferases catalyze the transfer of the amino group of an amino acid to an alpha-keto acid to form a new amino acid and a new keto acid. Aminotransferases are present in the liver, kidney, and the brain. Each enzyme is named in terms of 2 amino acids involved: the semaglutamic element transaminases, pyridoxal phosphate PLP, and the coenzyme form of vitamin B6 forms essential part of the active site of transaminases. Glutamic acid plus pyruvic acid in the presence of glutamic alanine and transaminase pyridoxal phosphate gives out alpha-ketoglutarate acid plus alanine acid. Another example of transamination is glutamic acid plus oxaloacid. Glutamic aspartate transaminase by pyridoxal phosphate gives alpha-glutaric acid, aspartic plus aspartic acid. In almost all instances of transaminations, glutamic acid is one of the reacting partners. A specific aminotransferase promotes each specific transamination. Right? The alanine aspartic amino acids are the most abundant. That's it for this session.